we had started a bridge course, and this bridge course is one of a bridge that connect your knowledge which you have taken in your graduation in BSc, and now you are forwarding towards the MSc. Okay, so the connection link between the UG and PG means from BSc to MSc that we have built in this particular course. Most of the topics that you have studied already in your graduation, but some of the things that it necessary to get to recall, because due to some this pandemic and the other things, some of the knowledge that we had taken in our previous colleges, I think we need to refresh it again. Yes. Okay. So I welcome you all in our today's lecture, and also wish you a very, very happy new year. Now we are in the phase of 2021. Okay. So let's take a look to the base topic. Okay. Today I'm going to deliver a topic, and that is, okay, I am Avinash Darsimbe. I'm working as an assistant professor in Department of Botany, Shivaji Science College of Botany. And today we will focus on one of the most important topics that had been asked in many competitive examinations like SAT, NET, GATE, JAM, various examinations. Most of the examinations, they cover the questions on this particular topic, and that is the cell cycle. So, dear students, before going to start to a particular cell cycle, we must know about the cell also. Okay, as in your previous lecture, you have studied that there are various type of cell organelles in a cell. Now, first we should know what is a cell. Okay, what is the structure of the cell? Actually, if we classify the cell, then basically there are two types of cells. One is a prokaryotic cell and another one that is the eukaryotic cell. Prokaryotic cells, they do not have the prominent nucleus and they do not have the bonded organelles. While the eukaryotic cells, they are the modified cells, they have the prominent nucleus and they have the bonded organelles. Now, if we classify the eukaryotic cells, then these eukaryotic cells, they are basically of two types. First one, that is the animal cell, and the second one, that is the plant cell. Now, in animal cell, in the plant cell, there is one major difference, that is the animal cell, it do not contains the cell wall, while in the plant cell, we can see here is the presence of the cell. Along with that, there are so many other differences also, like in animal cell, there is the chloroplast is absent, while in the plant cell, the chloroplast is present. Okay. So, it is a particular type of cell, we can say, and in cell, there are different type of organelles like Golgi apparatus, vesicles, microtubules, mitochondria, plasma membrane, lysosome, ribosome, microfilaments, centrioles, nuclear envelope, nucleus, and the nuclear core. In animal plant cell also, we can see that there is a presence of one of the thing that is not in the animal cell, that is a cell wall. It has the mitochondria, microtubules, ribosome, nucleus, rough endoplasmic reticulum, smooth endoplasmic reticulum, Golgi apparatus, chloroplast, vacuole, and plasma, and so many other organelles are also present. Some are very minute organelles, some are big in size, like vacuole, it is a very large organelle. Now, but as we know that this cell, they have certain different properties also. Like if we talk about the totipotent cell, means there are different potentials in the cell. There is a totipotent, pluripotent, monopotent cells are there. If we talk about totipotent, then we know that this cell have a property that it can divide and regenerate. So if it have the totipotency, then it can form a complete organism also. Okay, it means what? There are different properties of the cell more precious. And because of these properties only, there is a living organism only. So that the cells, they are the BC form. And these cells, they get modified. There is a different stages like morphogenesis for the formation of different type of organs. So all these things happens, but there is a particular mechanism for it. And that mechanism for the division, for morphogenesis, and other things that the cell can perform, for that there is a particular mechanism, and that is known as the cell cycle. Whether it is a cell or it is a eukaryotic cell, each and every type of cell, they have to go through this mechanism of cell cycle so that they can divide. There are various steps, and by these steps, these cells, they can divide and they can perform their different functions. So, in our today's lecture, we are going to talk about the various types of the particular steps that the cell have two phases while the process of cell division. Okay, and that is actually known as the cell cycle. Okay, so we are going to the topic, but before that, we just understand, if we don't know what is the cell cycle, if we are not studying anything, even though what naturally happens in the cell, this can be seen by this picture. Okay. In this picture, it is a realistic images that have been made just an animation, which can shows if we do not know what it is, if we don't know what happening in this, we do not know what happening in this cell. But these phases had to be covered by the cell while the cell has to be divided. So we just take a look to a particular first image. What it is, if we think it is a particular round shape, something is inside it, there are something is at the upper end. So it's such a, and here also we can see some type of we can say a globule-like structure is we can see. Now, then we identify that it is a particular cell. It is a cell which have a particular cytoplasm in which it have a permanent or prominent nuclei. Okay, and there is a presence of some type of fibrils or cantopores we can say at the upper side. Now, here is a particular inside the nucleus, as we know that there is a presence of DNA material. We can also say that it is a genetic material. Now, here it is in a thread-like form that is known as the chromatin nucleus. We can say it is a chromatin. Now, actually, it is one of the first image. After some time, you are watching that the same image, then it get changed to such different structure. Okay. Now, here we can see this structure has been changed. What's happening? Here, the genetic material is in a form of thread or fiber-like structure. Now, here is a formation of chromatids. Okay. And here we can say the chromatids, they have been cell. Now, likewise, here we can see also there is some changes in this particular cell. Now, again. Now, what's actually happening? Here, it is one of the matured cell. By the different stages, we observe that we just observing. Okay, we have not know anything about that. We are just observing a cell for a particular duration, about for 1 or 2 days. Then the changes, what happens actually? What actually we will observe in that particular cell, the changes what we observe, we are looking for that. Okay. So after some time, the particular cell change in this form, then after some time it changes in this form, then this we can see, and slightly we can observe at some stage came where there is the formation of such type of groups. Okay. Then after some time the groups get major, there is a shrinking in the middle portion. And finally, we found that, wow, it's amazing. It means what? First, there is a single image, and now we can see there are two different images of the similar type. Just like this image, we can see, oh, there is a two different type of images. It means what? There is a division in a cell. There is a division in a cell. It means what? First, there is a matured cell, And after some phases, we observed that from these particular single cell, we found two different daughter cells. Different daughter cells, okay? So from one matured cell, we found two different mature daughter cells. It means what? Something happening inside the cell which makes them to get divided. And this process, after studying, we found that it is a cell cycle. Okay, so generally we had just taken an observation of the cells. If we are not knowing anything, we are just observing the cell under the microscope, then which type of different structures, which type of different changes we can see in this particular cell. Now, we are going to study that. What actually happens? What happens in these cells so that they get divided? Okay. So, at this concept, it was then coined as a cell cycle. This concept, after studying this concept of dividing of the cell, it has been named as a cell cycle. Okay. One thing you are just, I think you are going to know that there are two different methods for the division of the cell. That is, first one that is mitosis, and the second that is the meiosis. Mitosis actually happens for the somatic cells only, and meiosis it happens for the germ cells. Mitosis, from one cell, the two daughter cells will form. In meiosis, from one matured cell, four haploid cells are formed. Haploid and diploid means what? Haploid, if you are talking about the haploid, it means it have n number of chromosomes, means half number of chromosomes. And if you are talking about the diploid cells, then they have the double number of chromosomes. It means 2n number of chromosomes. There is a presence of diploid number of cells, means 2n number of cells, and n it refers to the number of chromosomes. Okay, so this is the one of the basic things that you should know before studying the cell cycle. Now, in this particular image, you can understand there are two phases. First of all, the first phase, actually it is a cycle. We can say it is a cycle. In this cycle, there is a presence of two phases. First phase, it had been demonstrated by the black belt. Okay, it is the black belt which demonstrates the one phase, and the second phase it has been demonstrated by this red belt. Okay, so it is one of the phase, first phase, and it is the second phase. The first phase, it is known as the interphase, which is known as the interphase. Okay, and the second phase of the cell cycle, it is known as the M phase. It is the M phase. Now, in this interphase, it had been divided into three different phases again. Okay, the first phase, that is the G1 phase. The second one, that is the S phase. The third one, that is the G2 phase. Okay, so again, these interphases have been divided into three different phases, that is the G1 phase, S phase, and G2 phase. While talking about the cell cycle for the today's lecture, I am going to deliver the concepts of these are based for the particular categories. The main goal of today's lecture is just to understand the particular the events that had been taken in this cell cycle. Okay, so as concerning the eukaryotic cell, we are going through this, and in eukaryotic cell there are two main phases, that is the interphase and the M phase. The interphase, it consists of the G1 phase, S phase, and the G2 phase. Now sequentially we will go through the phases. First of all, we will talk about the G1 phase. Okay, now what is the G1 phase? Actually, in this phase, in G1 phase, as we had understand in the previous slide, that there is the formation of two daughter cells at the end. Now, consider that the one of the daughter cell is here. One of the daughter cell is here, means just it had been divided and had been placed, or we can say entered the G1 phase. Now, these daughter cell which had been entered in the G1 phase, it have a nucleus, and inside the nucleus it have a chromatine. Now, these particular new daughter cell, it starts to produce or synthesize its proteins. These proteins are required for the growth and development of the cell. So ultimately the cell starts to grow. Now here we can see if the cell is in this phase, at the starting phase of the G1 phase, it starts to produce their proteins. Means the machinery of protein synthesis, that is the translation mechanism. As we know that there is a transcription and translation. Transcription is for the formation of RNA from the DNA, and translation, it means from the mRNA there is the synthesis of protein molecules. Now, as from this particular stage, the cell starts to go towards this next phase. At the same time, if there is any abnormality in the cell, if the cell is not normal, then this cell go to the G0 phase. This cell go to the G0 phase. This phase, we can say it is just like changing, we can say. If your dress is not proper, you want to get ready. For that, you move to another room, and here you get ready for the next program. Now, here, if the cell starts the function normal, if the cell get normal, then it get again entered into the G1 phase. Okay, so in G1 phase, what happens actually? One of the cell, which is the daughter cell, it get entered. Now, in it, the synthesis of protein it gets started, cells start to grow, and if there is no, some abnormality in the cell, it goes to the G0 phase, again becoming normal and come to the G1 phase. Okay, so two events have been happening in this G1 phase. In G1 phase, the first one, that is the protein synthesis, and the second, that is the cell growth. Now, the cell growth means what? Cell growth, the definition of growth, it means what? If there is increase in size, mass, or volume of a particular substance, then it is known as the growth. And the growth, it is an irreversible process. It means what? If something get grow, then there is no another mechanism to get the growth reversed back. Okay. So cell growth is in which this cell, it started to increase in its size, in its mass, and its volume, and there is no any reversible step to get to we can say back to the another earlier phase. Okay, so now here is, we can say, the cell starts to grow, he become big. Okay, he starts to go towards the maturity. Now, so first there is a G1 phase. Now, after the G1 phase, when the cell is getting to be mature, it has all the proteins which are needed for its growth and development. It also increases its size, but there is a checkpoint. There is a checkpoint. You can see here is a checkpoint. That is known as the G1S checkpoint, which is known as G1S checkpoint. It means what? A checkpoint between the G1 and S phase. Okay, a checkpoint between the G1 and S phase. Now, in this G1S phase, there is a particular, in G1S phase, there is a particular checkpoint. It is known as the G1S checkpoint. Now, what is the checkpoint actually? Checkpoint means what? Just like a check post. Okay, if you are traveling from one place to another, then there are certain check posts are there, like the toll knockout. So this toll knockout, what is the actual purpose of this check post or toll knockout? That it will check whether you are going properly or not. Okay. Likewise, this checkpoint is to check out whether the cell have all the proteins that it needs, whether the cell grow or not. If it is not growing properly, if it have or not have all the proteins properly, then the cell will get restricted at this G1S checkpoint. Means the cell gets stopped, and no further development of the cell will take place. And if the cell have all the proteins, and if the cell is growing properly, then only then the cell will pass from G1S checkpoint and enter into the S phase. Okay, so if one of the daughter cell first starts to synthesize protein, then it grows, and all the machineries that it needed it will have, then only then it will pass this GNS checkpoint. Now the cell get entered into the please mute yourself, okay? Everybody mute yourself. Now, as the cell get entered into this interface, now the second machinery that is needed for the growth of the cell, and that is the DNA replication. Now here the proteins which are needed for the DNA replication, they are synthesized at G1 phase. Now these proteins will help for the replication of DNA. And DNA replication means what? There is a multiplication of DNA. Means number of DNA will start to form. Okay. Now, so in S phase, what happens? There is the replication of DNA starts. Now the DNA will start to form in a huge amount. Okay. So these particular S phase, it is for the replication of DNA. And this replication of DNA, it's a signal for a particular cell that now you are capable to move forward for the next phase, and that is the G2 phase. Now, as the cell crosses the S phase, it starts or it gets entered into the next phase, and that is the G2 phase. Now, in this G2 phase, what happens? The final protein synthesis means what? The advanced proteins which are needed for the further development of the cell, for the further growth of the cell, they had been synthesized. So, as the previous type of proteins that will form in the G1 phase, now the advanced type of proteins and which are needed for the further development that will form in the G2 phase. So, the final protein synthesis of the cell, they had been synthesized in the G2 phase. Okay. Now, as all the proteins they had been synthesized in G2 phase, then and then only it moves forward, and there is the another checkpoint, and that is known as the G2M checkpoint. Now, at this G2M checkpoint, the cell again gets checked. It is a checkpoint. Means again, it had been evaluated that whether the proteins that are needed for this particular cell, which are required for this particular cell for the further development, whether these particular proteins had been formed or not. If these proteins had been formed, then and then only the cell will pass this G2M checkpoint, because it is a checkpoint between the G2 phase and M phase. That's why it is known as the G2M checkpoint. Now, if the proteins that had been needed for the particular development had been formed, then and then only the cell will pass from G2 to M phase. And if the proteins are not formed properly, then from this phase, the cell will get restricted, means the development will get stopped. So, if the cell passes the G2M checkpoint, then it will enter into another phase, and that is the M phase. It is known as the mitosis or meiosis phase. That's why it is known as M, we can say. Okay. So it will be a mitosis phase or it will be a meiosis phase. If it is a somatic cell, it will be a mitosis phase. If it is a germ cell, then it will be a meiosis phase. Now, but in the interphase, there is only the formation of particular machineries and proteins which are required for the division and for the growth and development of the cell. Now, the actual division of the cell, it starts in the M phase. The actual division of the cell, it starts in the M phase. Now, in M phase, if we are talking about the mitosis, then we all know that it have the 4 different phases. And these are, the first one that is the prophase, the second one that is the metaphase, the third one that is the anaphase, and the fourth one that is the Telo phase. And in this particular phase, in anaphase and Telo phase, it has been followed by the process, and that is known as cytokinesis, in which the cytoplasm of the cell starts to divide. And after this, the cell will form a 2 daughter cells again, and it will again into the next phase of the particular daughter cell, that is the G1 phase. Clear? So it means what? If the cell passes from the G2M checkpoint, it will enter to the mitosis or meiosis phase. And in these phases, it will first enter into the prophase, then metaphase, then into the anaphase, then into Telo phase. And there is a cytokinesis of the cell, means division of the cytoplasm, and we can finally found 2 daughter cells as we had seen in our previous image. And one of the daughter cell, it will again enter into the G1 phase. Again there is the interphase with G1 as G2. Then again there is a division in M phase, and again we will form the two daughter cells. So this is a cycle. It is continuously take place by the cells simultaneously. So as we have seen that how a particular daughter cell, it enters into the G1 phase, how it crosses the G1S checkpoint, how it enters into the S phase, then G2 phase, then G2M checkpoint will get crossed, and in M phase how there is a division, and finally again it is a formation of one of the daughters. Okay, so it is one of the brief which makes a scenario how there is a changes in the particular cell and how it will get. Okay, am I audible? Am I audible? So let's take a look which what are the phases of the checkpoint of the cell cycle. As I told you in the earlier slide, that first there is a G1 phase where the metabolic changes, then prepare of the changes, prepare for the cell will prepare for the division. Then in G1S checkpoint, at a certain point, the restriction point, the cell is committed to division and move into the S phase. That is it is one of the checkpoints G1S checkpoint. Then in S phase, the DNA synthesis, replicate, and the genetic material. Each chromosome now consists of two sister chromosomes. Now the DNA synthesis takes place, the genetic material gets replicate, and now the two sister chromosomes they are. In G2 phase, the metabolic changes assemble the cytoplasmic materials necessary for the mitosis and cytokinesis. So in G2 phase, the materials which are needed for the mitosis and cytokinesis, they have been gathered. Now in the G2M checkpoint, again there is a cytoplasmic material formed or not, it has been checked out. The cytoplasmic material which are needed for the photo development, whether they are formed or not, it has been checked. Now in M phase, a nuclear division, that is the mitosis, followed by a cell division, that is the cytokinesis. Okay, one thing you must know that the division of the particular cell, it takes into two phases. The first one, that is the division of the nucleus, and the second phase, that is the division of the cytoplasm. Division of the nucleus, it comes on the phase, and that is the mitosis or meiosis. In this phase, when the cell get divided, then the chromosomes they also get separated. And this particular event, which is the division of the nucleus, it is also known as the karyokinesis. Karyon, in Latin word, it is known as the nucleus, and kinesis, it means the division. That's why it is also known as the karyokinesis. Likewise, cytokinesis, cyto it means cell, and kinesis it means division. When there is the division of the particular cell, or cyto, we can say there is a cytoplasm actually. Cyto it means cytoplasm. So when there is a division of the cytoplasm, means cyto means cytoplasm, kinesis means division, then it is known as this. That's why it is known as the cytokinesis. Okay. So these are the particular phases of the cell cycle. Now, as the main events, that is the G1 phase, G2 S checkpoint, S phase, G2 phase, and G2M checkpoint, and M phase, we now understand. Now we'll move to the particular event that takes in the cell, and these are the mitosis. How these particular cells get divided, we can understand it by the mechanism of the mitosis. Now, what happens? Now we are entering into the M phase actually. Okay, we have processed the interphase. Now we are on the M phase, and if we are considering a somatic cell, then we will understand, we must know, or we can consider that it is a mitosis. Now, in mitosis, there are first one, that is the prophase. In prophase, there is the two different phases are also there. That is the early prophase and late prophase. Okay, early means at the beginning, late means at the end. Okay. Now, after prophase, there is a metaphase. After metaphase, there is an anaphase. Then there is a Telo phase, and from the anaphase, the process of cytokinesis gets started, and at the end of Telo phase, there is the division of the cytoplasm. So it is a particular steps. Now we will understand each step in a detail. Okay. So first we will talk about Actually, when we are talking about the cell cycle, we are talking about the particular interphase and the end phase. Means both the cycle, both the particular phases. Means if we are talking about the cell cycle, we must know the interphase also means G1H, G2M, and G2 and M phase, that is the mitosis of cells. Now, the first step of the mitosis, that is the early prophase. Now, here we can see it is a cell, and in this cell there is a particular nucleus, and in this nucleus there is but if at the early phase, we can say early prophase, actually there is a presence of the chromatin only. Now, as the time passes on, these chromatin starts to condense, and after condensation of the chromatin, it forms the chromosomes. Now, what happens as this nucleus is prominent at the primitive stage? Now, this nucleus and the membrane around the nucleus, that is the nuclear membrane, it starts to disappear. So the nuclear membrane starts to disappear in this early phase. Now here we can see there is a presence of these spindle fibers, chromatins actually. So from these kinetochores, these are the kinetochores, the formation of a spindle fiber, it gets started. Okay, so what happens actually in the early phase? The events what happens, the first one, that is the chromatin starts to condense. The thread-like structure of the DNA is known as the chromatin. When you look to a particular nucleus, inside that there is a thread-like structures. The thread-like structure or ribbon-like structure of the DNA, it is known as the chromatin. Now, as the chromatin starts to get condensed, it starts to form the chromosome. Means this chromatin get converted into another form, and that is known as the chromosome. Okay. So this process, it gets started in the early phase. Now, second thing, the nucleus and nuclear membrane starts to disappear. Now, as the nucleus, it is a prominent organelle and has a nuclear envelope, means there is a double membrane structure around the particular nucleus. It is known as the nuclear membrane, and these nuclear membrane now it starts to get disappear, and nucleus it also starts to disappear in early phase. Now, the third event that happens, that is the spindle fiber begin to appear. Means here we can see from this kind of course there is a formation of spindle fibers. These spindle fibers like fiber-like structures. Okay. So if these are the two different poles, then these spindle fibers they are start to form. It's a fiber-like structures. Okay. So such type of fiber-like structures have been started to form in the early phase, and it is known as the spindle fibers. And these spindle fibers, they are the actual carrier of the chromosome from the particular location towards the pole. Okay. These are the spindle fibers which are attached on the chromosome, and they carry this chromosome to the different poles. Okay. You will see it on the next slide. Now, so it is the early phase. In early phase, there is a three events happen. What are these three events? These are the first one, that is the chromatin starts to condense. Then the nucleus and nuclear membrane starts to disappear. Then the spindle fiber begin to appear. Okay. Now the next phase, and that is the late prophase, or it is also known as the pro-metaphase, because after prophase there is a metaphase. Okay. So late prophase, it means what? When the cell enters to the end point of the prophase. That's why it is known as the late prophase. Okay, that's what is known as the late phase. Now, and it is also known as the pro-metaphase. Now, what actually happens in this particular phase? As we know that in early phase there is the start of the condensation of the chromatin. Now, these chromatin they get totally condensed and they form the chromosomes. Here you can see there is a structure. Okay, there is a particular structure of a chromosome. Now, these chromosomes they get appeared by the formation of, or we can say condensation of the chromosome. So first event that happened, that is the chromosome forms. Now the second thing, that is due to growth of the spindle fiber, the centrioles start to move towards the opposite pole. Now From here, it is a one of the pole and it is one of the another pole. From this pole, there is a formation of spindle fibers. These spindle fibers are starting to get attached to the chromosomes. Okay. So, in late prophase, there is a formation of, or we can say elongation of these spindle fibers. They start to get elongated. Now, next one, that is the kinetochore from each chromosome move towards the opposite pole. Now what happens? These particular spindle fibers, they are going to attach to the chromosomes and they will start to move towards the opposite poles. Okay. Some of the spindle fibers, they will try to push the chromosome, and some they will start to get attached to the chromosome. They will carry towards themselves to the particular pole. Okay. So in late prophase, there are three events again. These one, the first one is the chromosome get formed. Now the second is due to the formation of spindle fiber, the spindle starts to move towards the opposite pole. Then the kinetochores from each chromosome move towards the opposite pole. Okay. Now, so these kinetochores are moving towards the, from this pole they are moving towards the this pole. Okay, or we can say spindle fibers. Now here it is the image which shows the particular anatomy of the spindle. Okay. Spindle means what? These are the spindle fibers. Now, this particular pole, there is a formation of esters. Okay? These are the esters which are moving towards the opposite direction. Okay? These are the spindle fibers, or we can say they are known as the kinetochore microtubules. Now, here we can say there is a chromosome, and in this chromosome, if we are enlarging this particular image, then we can see there is a presence of the kinetochore here, and here it is the centromere. Okay. Now these kinetochores, they are attaching to these kinetochores, they get attaching to these particular centromere. It means what? They will attach to the centromere. This kinetochore, this microtubule kinetochore, they will attach to the particular centromere, and from the both sides they get attached, they will start to separate this particular two different chromatids. These are the two chromatids of the chromosome. It is one chromatid, it is the another chromatid. Okay. So the chromosomes, they will get separated from each other by the action of these kinetochores. Now the next phase, that is the metaphase, and it is one of the most observable phase if we are doing a practical of any type of tissue or any type of cell for observing the mitosis or meiosis. In that, you can easily understand the metaphase. In your 12th class and in your graduation also, you have done the practicals of mitosis and meiosis. Okay, I think from the onion group tip, you had already performed the practical of the observation of mitosis, different stages of mitosis from the onion group. Okay, so what actually happened in this metaphase? In metaphase, we can see here all the chromosomes, they are aligned at the center. All the chromosomes, they have been aligned at the center, which what? The chromosome line up at the metaphasic plate, and this plate is the central point, it is known as the metaphasic plate. So again, three events that have been happened in this metaphase. That is the first one, that is the formation of the spindle fiber completed. Now the spindle fiber, they have been completed totally. There is no another development in this spindle fiber take place. They have been completely formed. Second thing, that is the chromosome becomes still shorter and thicker. Now those chromosomes, they are becoming more shorter and more thicker. Means more condensation happens in this particular type of chromosomes. The next thing, that is all chromosomes are aligned on the equatorial plate. All the chromosomes, they align on the equatorial plate, and that is known as the metaphasic plate. So in particular metaphase, we can see all the chromosomes, they are aligned on the center, or we can say the equator, which is known as the metaphase. So this is the metaphase. After metaphase, the next step comes, and that is, we can say, towards the starting the division of the particles. So the next phase, that is the anaphase. Now this anaphase, it had been also divided into two phases, that is the early anaphase and the late anaphase. Okay, it also had been divided into different phases, that is the early anaphase and the late anaphase. Now what actually happens in the first phase, and that is the early anaphase? Actually, as I told you, that it is the starting of division of the particular cells. So in this phase, the separation of the chromosomes gets started. So what happens actually in early anaphase? It is also known as the separating phase. It is also known as the separating phase. In this phase, as you can see that here it is one of the pole, it is the another pole. Now these chromosomes, they get separated from each other. They get separated from the each other. Means what? From the particular their centriole, they had to get divided. So what happens? These microtubules, they are pushing them pull or towards the pole apart, and the kinetochore microtubules pulls the chromosome towards the pole. Okay. So some of the microtubules, they are pulling these chromosomes towards the downward side, and the microtubules of the upper pole, they are pulling this chromosome towards themselves. So because of that, these chromosomes, they get separated. Okay. So the two sister chromatids of each chromosome get separated, means we know that now the sister chromatids that get separated. Okay. Now the next thing is that and start to move towards the opposite pole, and they are starting to move towards the opposite pole. So some are moving towards the upward direction, towards the upper pole, and some are moving towards the lower. The next thing that is the UN, and that is the late N. It means what? That is the completion of the anaphase. And at the completion, if you are talking about that, it means what? The things that had been started in early anaphase, they will get completed in the late anaphase. So in late anaphase, what happens? These separated microtubule, they get segregated to the opposite pole. Means if we are seeing in this image, they are in only placed in the middle of the cell. Now in late anaphase, they will get segregated at the particular pole. So all the chromosomes will get segregated at the pole only. Here also all the chromosomes, they will get segregated at the pole only at the late anaphase. Means total separation of the chromosomes will get takes place. So that is the anaphase. Now as we had completed the anaphase, the last phase of the cell division, or we can say mitosis, comes. Actually the last phase of the nuclear division, we can say. And that is known as the Telo Phase, and that is known as the Telo Phase. Now, actually, in Telo Phase, these Telo Phase may also get divided for our better understanding into the two different phases, that is the early Telo Phase and the late Telo Phase. Now, as we know that already the chromosomes, they had been pulled towards the two different poles, means the separated chromosomes. Now, in this particular step, that is in Telo Phase, what happens? The chromosomes aggregate at opposite pole and get elongated. Now, as we know that the chromosomes have been segregated at the opposite pole, and in metaphase we know that at that phase there is the more condensation of the chromosome takes place. Now, these chromosomes, they are trying to get elongated. They are going to get free. So the second thing happens, that is the chromosomes are going to elongate. It means they are coming to its original. The second thing, the spindle fiber begin to disappear. Now these spindle fibers, in last slide we see that there is the spindle fibers from these poles. Now these spindle fibers, they get totally disappear. There is no spindle fiber over here because these spindle fibers, they had already performed their duty. They had taken this chromosome to the opposite poles. Now they had finished their duty, they had completed their work. That's why they get disappear. So these spindle fibers, they get disappear in this early Telo phase. The next thing is, the nucleus and the nuclear membrane begin to reappear. They begin to reappear. It means what? As the separation of the chromosomes takes place to the different poles, now there is a need to cover these chromosomes. And for the covering of these chromosomes, there is the formation of nuclear membranes. Now again, this nuclear membrane, which has been disappeared in the prophase, early prophase, it starts to reappear. Means the reversible cycle has been started. Now, so here you can see there is a formation of the particular nuclear membranes takes place. The chromosome, they get separated to different opposite poles. The macrotubules, or they have been get disappeared. Now the last step, and that is the late Telo phase. In these late Telo phase, what happens? The two daughter nuclei form at the opposite poles. Finally, when the two different particular portion, they have the nuclear envelope, then we can see that there is a formation of two different nuclei. There is a formation of two different nuclei. So here is the one nuclei and here is the two nuclei. When we are talking about one of the matured cell at the start, it has only one nuclei, but these particular cell have the two nuclei. It is known as the dinucleate condition. It is known as the dinucleate condition. The cell had not get divided. Only a single cell, they get separated its nuclei, or we can say division of the nuclear takes place. That's why it is known as the dinucleate condition. Okay. So now, two daughter nuclei that have been formed. Now each daughter nuclei, it contains the equal number of chromosomes. Each daughter nuclei means this one and this one, both the nuclei, they have equal number of chromosomes. Means the equal separation of the chromosomes it have been takes place. So now we can see here there is the formation of two different nuclei, and we can say it is the last phase where our nucleus it gets separated. I already told you that in M phase there is prophase, metaphase, anaphase, and Telo phase, and in M phase if we divide this phase into two parts, then first there is a nuclear division and then there is a cytoplasmic division. The nuclear division is actually known as the mitosis, or it is also known as the karyokinesis. And after the separation of two different nuclei, there is the separation of the cytoplasm. Here you can see it is a cytoplasm, it is a cytoplasm, it is a cytoplasm. Now the next step will come where this cytoplasm it will also get separated. Then we will get two different cells, and the separation of two different nuclei, or we can say the separation of particular cell through the cytoplasm, it is known as the cytoplasm. So the next step, that is the cytoplasm. Now, so after in the M phase, at the end, there is the division of the cytoplasm, and it is known as the cytokinesis. But this cytokinesis it happens in two different manner, in animal cell and in plant cell. Okay, if we are talking about the animal cell, then you can see these particular animal cells do not have the cell wall, and because it do not have the cell wall, that's why the shrinking of the particular cell from the outward to the inward side takes place. Okay, so division starts from the outer side. Okay, so in animal cell, when division starts from the outer side, it is known as bi-furrowing. It is known as furrowing. The method is the furrowing method. Okay, as we say that the cytokinesis is known as the division of the cytoplasm. Cytokinesis is known as the division of the cytoplasm. In animal cell, these divisions take place by the formation of furrows. By the formation of furrows, it is known as the cleavage furrow. It is known as the cleavage furrow. Okay, and there is a ring formation at the center, and it is known as the actin ring. It is known as the actin ring. And finally, when this ring it form and the cleavage it get deeper, it will separate it into two different cells. So we will get two different cells. Okay, so it is the cytokinesis in the animals. Now, something has been changed in this particular plant cell. Actually, what happens? If we are talking about the plant cell, then this plant cell, they have the cell wall. They have the cell wall. And because of the presence of cell wall, the particular furrow formation from the outside, it will not take place. So instead of that, the formation of these separating plates will take place inside the cell, and it is known as the cell plate. From the inner side of the cell, there is a formation of cell plates, and it will start to grow towards the cell wall. So here, when the cell plate will get formed, then this cell plate will grow towards the cell wall, and from the inner side, there is a division of the cell. Means division of the plant cell, and these division of the plant cell from the inner side, it is known as cell plate formation. So in plant cell, the cytokinesis will take place by the cell plate formation. Clear? So the cell plate formation will take place. So now we can understand the cytokinesis in animal cell, in plant cell takes place by two different methods. In animal cell, this division takes place from the outer side of the cytoplasm, which is known as bi-furrowing, and in any plant cell, the division takes place from the inner side of the cell. It is known as by the cell plate formation. And finally, both animal cell and plant cell, after the cytokinesis, will get two different cells. Clear? So here we can see finally we get two different cells, and it is a complete method, or we can say phases through which a cell it get divided. Clear? So as we had seen our first image, I just want to take a look of you to the particular image that we had seen in the previous. That is how this particular single cell, it goes through different phases and finally it get entered, or we can say it can appear as two different cells. So at the end of the cytogenesis, we will get these two different cells. Clear? So these images shows how these particular matured cell go through the cell cycle, and in cell cycle there is interphase and M phase, and finally it gives two daughter cells. If you are talking about the mitosis. Clear? So I hope that you will understand, all the things that I want to deliver to you. And if you have any query regarding this particular cell cycle, you can ask. Okay. So I think you must ask some of the questions that are raising inside your mind. So if you have any question, please ask. So any questions are there? About 23 students had been joined for the today's lecture. So if anybody have any query regarding this cell cycle, the topic is now open for the discussion. Okay. So if you have any query, you can ask. There is one of the famous phrase in the field of education, and that is no study, no conclusion. More study, more knowledge. Okay. So in which particular line you belongs to, it must be we can say observed by yourself. Okay. So if you have no any doubt, it means whether you understand everything or you can't understand. So is there any question? Is there any question? You do not have any question? I think each student must ask one question for the particular topic. Come on, Bhavna. You have any question? Sakshi, Janvi, Anushka, whether you are watching the particular lecture or not, आपने lecture सुना कि नहीं सुना?